It's not even September, and I felt fall in the air the other night, Steve. I don't want to say it, but I think that's what it was. And of course, it was gigantically humid and thunderstorms the next day. So,
Wait, you can feel pumpkin spice?
oh no, did you say that word?
I did. You said fall, and you could feel it. So that must mean you can somehow feel the pumpkin spice in the air.
I could, and I think everyone who's listening right now just smelled pumpkin spice. I think this is about the worst thing we could do to anybody. It's going to be a fun show today. New news about a multiple sclerosis treatment that's not a drug. We'll get to that in just a minute. Also, an amazing interview with Tony Eisenhut of NovaSterilis. We'll be talking with him all about sterilization techniques, novel sterilization techniques, really cool stuff, as well as upcoming events from the local and national ecosystem up at the end. All this next up on MedTech Crossroads. Multiple sclerosis, abnormal sensations, muscle spasms, stiffness, weakness, mobility issues, pain. I have a friend with the condition. It's nothing at all to be laughed at, and many people suffer from this condition. Well, it turns out that August 24th, yesterday, FDA approved. It's not a new drug. It's not a new device. It's a biologic for relapsing multiple sclerosis. And I thought we'd dive into this just a little bit today. What is a biologic? Many people have not heard about biologics. They know what a drug is. They know what devices are. We work on devices all the time over here at Into Being. But what's a biologic? Well, if a drug takes some natural material and reduces it down till it becomes this one isolated thing, a biologic starts with living cells or microorganisms, and it makes the product out of living cells or microorganisms. It makes it from those. And this then leads us to the idea of what is called a biosimilar. So if I make another set of this, unlike a drug, where I could make the identically same thing, in the world of biologics, this is not going to be identical. Whatever I make over here is not going to be identical as the thing on the left. And that's why we call this a biosimilar. Now, you may already be aware of this, but these are regulated by different agencies at the FDA. So drugs are regulated by the CDER, Center for Drug Evaluation and Research. Devices that we work a lot with at Into Being, CDRH, Center for Devices and Radiological Health. Biologics covered by CBER, Center for Biologics Evaluation and Research. And so what we have today is the first biosimilar, not the first biologic. There was a biologic cleared that we'll talk about shortly. We're not talking about drugs today. We're talking about biologics. And what just happened yesterday was the first biosimilar that has the same indications. It's treating the same thing as this biologic that came out. But now there is another one on the market, and so it's called a biosimilar. So from FDA, both a biosimilar and its original biologic are made from the same types of sources. Those are living sources or microorganisms. They provide the same benefits when treating diseases or medical conditions. They're given at the same strength and dosage, and they're not expected to cause new or worsening side effects. Some of the things that biologics are used for: chronic skin diseases such as psoriasis, chronic bowel disease such as colon, Crohn's disease, and IBS, diabetes, macular degeneration, arthritis, kidney conditions, and some cancers. So biologics are making waves. FDA also says this: compared with an original biologic, a biosimilar is made with the same type of natural sources. It's given the same way. It provides the same treatment benefits, has the same potential side effects, We'll talk about that in a second, and has the same strength and dosage. The reality is, though, it can't be identical, right? In the world of devices and in the world of drugs, we can sort of guarantee, it's not identical when you make two devices. They're truly not perfectly identical, but for all intents and purposes, they are. And so a biosimilar is similar, but not identical. Nevertheless, it comes from the same thing, it's given the same way, has the same benefits, and is given in the same manner. Interestingly enough, before we go to the particular biologic that was released, insulin for diabetics, it was always regulated as a drug historically. But insulin is made in living cells. So insulin and other drugs that meet the criteria for a biologic are now regulated as biologics. This doesn't change the ingredients of insulin or how you obtain medication at the pharmacy. This is all from FDA. This is not our language. But insulin is now being treated, I guess as it should have been, as a biologic. So what's new? Well, this is the new biosimilar. It's called Tyruko. And this is the one by the company Sandoz Inc. that is for relapsing multiple sclerosis. Again, this is not medical or legal advice. Talk to your doctor if you have any thoughts about this. We're just fascinated by the clearance processes for these things that are neither drugs nor devices, but are biologics. Well, what is the original biologic upon which Tyruko is based? It's called Tysabri, and it's doing the same thing. It's for this type of relapsing multiple sclerosis. But it turns out that back in 2006, Tysabri was, during its clinical trial, was pulled temporarily before, it's February 2005, right here. It was withdrawn by its maker, Biogen Idec, and the distributor, Elan, after 3 patients in clinical trials developed progressive multifocal leukoencephalopathy. I think I got that out, which is a viral infection of the brain, a rare one at that, but 2 cases were fatal. So it turns out, as I was reading about Tysabri and about Tyruko, this is still a concern, but there have now been things done to reduce the likelihood that it would have this kind of an outcome. And so Tysabri being back on the market, Tyruko now being approved by FDA as a biosimilar, although with concerns about this PML outcome. And so new hope for people with relapsing multiple sclerosis. Again, this is not something that we're giving medical or legal advice, but we did find it fascinating that this new pathway at FDA, biosimilars, the ability to clear things that are not identical, but that are derived from living cells and other microorganisms. They can't be identically similar because they're new batches of stuff. And nevertheless, FDA is paving the way for things like that to make it to market. Well, if that was useful to you, we hope that you will like and subscribe. We hope that at MedTech Crossroads, what we offer here for free will help you save you headaches on your MedTech development journey. Ethylene oxide alternatives, gamma sterilization alternatives. What about low temperature sterilization methods? What about low temperature vaporized hydrogen peroxide, for which a consensus standard was recently recognized by FDA? What about new methods like supercritical CO2? Just a little bit earlier today, I was able to catch up with Tony Eisenhut, who is the CEO and co-founder of NovaSterilis. They're working on a novel sterilization technology, and Tony was able to unpack for me this new FDA news, as well as a deep dive into education about sterilization. I hope you enjoy it. NovaSterilis. Welcome, Tony.
Welcome. Thank you for having me, Gene.
It's great to have you. Thanks for joining us. You know, I think people think, oh, maybe they bake them, maybe they, but of course, with so many polymer devices, that often doesn't work. There's so many ways to do this. But first of all, what kinds of things even get sterilized?
You'd be surprised, but everything from Band-Aids to endoscopes that they use in hospitals and healthcare situations, up to therapeutics. Drugs that we take are often sterilized. Some of the regenerative medicine products that are being introduced into the market today are sterilized as well.
It's everything. It's not just the hypodermic needle that you think of as being that. It's sort of all these things. And Band-Aids too, you mentioned that. I mean, I guess we take them out of that little wrapper, and that's why they were in there in the first place.
Yeah, they do. I don't know if it's hundreds of thousands or millions of Band-Aids a day are being sterilized.
So how does this happen? I know we did a segment one time on the show, and I actually went to the lab and I took a syringe and I autoclaved it, as it were, and I brought it out and showed that, okay, not all devices can be sterilized just using heat, because it was a pile of goop at that point. What are some different ways that people sterilize things, maybe historically, and then some of the more novel ways that are coming out?
Yeah, sterilization has been around for centuries. Fire, right? You had wounds, I don't know, going back to the Dark Ages, and you'd use some fire, a piece of metal, and you'd cauterize a wound. Well, you were also sterilizing that particular wound. Clearly not a technique that's broadly applicable to medical devices today, but there's some of the workhorses of the terminal sterilization, especially on the industrial side for medical device manufacturers, pharmaceutical companies, regenerative medicine, have historically been ethylene oxide sterilization, often referred to as ETO, and gamma radiation. They probably make up about 95% of the total terminal sterilization market for industrial applications.
And this is something we didn't really talk about before, but I know something we've experienced in our development processes, and I think you could speak to a bit. Not all sterilization methods work on all products, partly because of the materials of the products. I remember working with gamma, and you can't use that plastic because it's going to embrittle it and it's going to yellow it.
Yeah, no, and so the workhorses, as I just said, were the gamma and the ETO, but more recently, and I say more recently, over the last 20 plus years, there have been novel sterilization modalities or alternative sterilization modalities that are coming forward, whether it's vaporized hydrogen peroxide or our modality, for example, supercritical carbon dioxide, hydrogen peroxide plasma sterilization, ozone sterilization. There have been a number of new technologies that have been developed over the time because exactly what you said, medical devices, therapeutics are getting more complex and more materials are being used. Sometimes combinations of, if you look at a drug-eluting stent, right? You've got a therapeutic, you got a polymer, and you've got a metal component all in one device, and that device has to be sterilized. And so, as you said, maybe gamma is not compatible with the polymer, EO might not be compatible with the therapeutic, and so alternative sterilization modalities are necessary today as the med device world evolves and moves forward.
And it seems to be picking up speed. Talk to us about this. I mean, I've noticed in industry, right? Like in academia, people love to innovate, and you've always got something new because we've got to write a new paper, and we've got to get P less than 0.05, right? So anything new we can possibly show, like we want to do it. But in industry, it's very much the opposite. Once we find something that works, do not touch it, do not let it move. But we're seeing this increased uptick in novel sterilization methods, and NovaSterilis, which we'll talk about as an example of that. Why the right now huge uptick in it? What's going on with these established processes?
Yeah, the interest has grown dramatically, and as much as we love the sterilization industry, it's not the most glamorous. It's a necessary evil in a lot of manufacturers' mind, and it's important. And I'm not saying that they don't value it, but if you're a manufacturing company, you're trying to grow revenue, and sterilization processes don't grow revenue per se in and of themselves. So if it works, people have historically stuck with it. But what's driving the change today is innovation in the products that are being brought to market, but there's also some regulatory and really clean tech concerns and safety concerns. For example, ethylene oxide. It's very effective, but it is a carcinogenic, mutagenic, toxic gas. And as EPA is looking at ethylene oxide and changing some of the regulations relative to emissions, the cost of ethylene oxide is just getting more and more expensive for them to be able to meet those standards. And with that being the case, companies are starting to look for alternatives. There was an incident in 19 where a couple of facilities got shuttered, one on a temporary basis, one on a permanent basis. And so the availability of ethylene oxide capacity was greatly reduced. Lead times got very long. And again, candidly, if you're a small company, you weren't really even getting a chance to get in the queue because the larger companies were taking up most of the capacity. The other issue is
as the capacity is dropping, the small ones have no chance of getting in at that point. It's just
it gets very expensive,
door.
and it just gets tougher to get in, and the lead times and the wait times get a lot longer than what they had been previously, and so. And then on the other front, in 2019, it was kind of a double whammy for the sterilization industry. When it comes to gamma, cobalt 60, which is required for gamma radiation, supply channels were tight. Cobalt comes from some of the more challenging geopolitical regions of the world, and as a result, the supply chain was a little bit tight, and that created concerns for people. Pricing went up, and what was the availability going to be? In fairness, some of that has alleviated, but it's on people's mind. It's still a fresh wound per se, and so they don't want to be in a situation with their sterilization supply chain where they can't put product into the market, because if you can't terminally sterilize a product that's required to be terminally sterilized, you can't sell it.
Yeah, you're dead on arrival there. You may have product sitting there, but if it's not ready to go, it's not ready to go.
Absolutely.
Well, FDA has ways of categorizing these sterilization methods, and I think these are fascinating. Unpack this for us as a technology is maturing. What are the categories that FDA has established for these sterilization technologies?
So really, there's two designations. There's the established sterilization modality, and then novel. And then within the established sterilization designation, there is Category A and Category B. And so Category A are the industry workhorses, the ones that have been around: heat, EO, gamma. There is a long history. And so they are well understood at the agency. Category B are newer modalities that have gone through more evaluation by the agency and have generally met some standard. And of late, it's been industry standards, whether it's ISO or other standards that exists out there. And so, for example, vaporized hydrogen peroxide, flexible chamber, ETO, ozone had moved from what was novel into established Category B. And then there's another category, which is the novel, and those are the newer modalities that the agency is getting their hand around. It doesn't mean, and one of the misconceptions we see often is that when people hear novel, they think, oh, it's not going to be able to get approved, or it's never been approved, or it's going to take a long time to get approved. And the reality is that's not necessarily the case. It's that the agency is looking for the same information that the other modalities have, but it's being generated on a per submission basis. And so, in our instance, and we're designated as a novel sterilization modality today, there were 4 clearances last year by device manufacturers who are using our modality, and were able to get their device approved by the FDA last year.
So the novel and established categories are not, like you said, a way of FDA saying, oh, that's never going to happen. It's actually a way of FDA acknowledging that there's a maturation process of these, and they're looking at it on a per submission basis for the novel ones, and then looking at it more established for the established ones.
Yeah, that's a good way to describe it.
I wonder why they picked those words. There you go.
and it's changing.
Oh, go ahead.
Well, it's changing from the agency's perspective, because they understand the supply chain risk that exists out there today. And so in 2019, they put together a program called the Innovation Challenge Grant Program. And it was, or is, the FDA's way of helping novel sterilization modalities or new techniques to move into the mainstream, to help the FDA understand them better, so people can switch from the, I'll say, category A established techniques into some of the newer modalities as required by device complexity and supply chain issues.
And I understand, we'll talk about this in a minute also, as well, is that NovaSterilis was a recipient of that challenge grant award. So congratulations. We'll dig into that in just a few minutes. This is a pre-novel step? These would be devices that are going to become novel, or these devices that are in the novel category?
Are you speaking about the sterilization technique or the devices themselves?
Oh, I was thinking the challenge grant stuff. That's all for like novel and pre-novel devices?
It is for it's for novel devices as well as established. So, for example, Noxalizer, NO2, nitrogen dioxide sterilization, is one of the recipients. And I believe they are still designated as novel, but not 100% sure on that. We are at supercritical CO2, but Steris was part of the program with vaporized hydrogen peroxide. That's in the category B established technique. They have Steris got a second one with radiation, and that is a category B established technique. And then Stryker, through their TSO3 platform, which is also an established category B technique, is a vaporized hydrogen peroxide ozone approach to terminal sterilization.
So FDA's effort there was independent of which category it fell into. We're going to come back to the category stuff, because I really want to spend some time with you on NovaSterilis technology and that award that you got as well. But I want to back all the way up for just a moment, because we're sitting here talking about sterilization, and I think it will come as a shock to many people that while, as you said a moment ago, terminal sterilization is recommended or required by FDA where possible.
Yes.
This isn't always the case. Like, not everything that we think, in our minds should be sterilized actually is. Tell us more about this.
Yeah, so the FDA's guidance is that if something can be terminally sterilized, they want it to be terminally sterilized. Now, if terminal sterilization will negatively impact the efficacy of the product or the effectiveness, then that's not a good path forward. And so in those instances, the FDA is saying aseptic manufacturing is the appropriate approach. And so, and this is a moving bogey to some regard, right? Because if you go back a decade, for example, tissue products were not being terminally sterilized. They were being aseptically manufactured. So if you go in for a surgery and you have an ACL replacement that's needed and you're using an allograft, that was most likely aseptically manufactured. But with new techniques that are out there today, there are terminal sterilization solutions for products like that. But also think of large molecule therapeutics, a number of those, because proteins are often not compatible with radiation or ethylene oxide, are not terminally sterilized. They're aseptically manufactured.
When you think of these things, I'm putting it into my body, it must be sterilized. And the answer is not quite. It might be aseptically manufactured. That's absolutely fascinating. Let's talk a little bit of people who get close to the sterilization industry, excuse me, keep hearing this 10 to the minus 6 number. Yeah. And it always is this magic number. Yeah. And we've talked about it on the show before, but unpack this for us from your perspective.
Yeah, from our so sterility assurance level of 10 to the minus 6 is representing that not only are you killing the microbial population of 6 logs, which is the overkill method, that you're doubling that condition. So theoretically, you're killing 12 logs of microorganisms. You can't grow 12 logs of microorganisms, so you really can't prove it. But from a mathematical perspective, by doubling the conditions, whether it's time or the concentration of additive, you are giving a much higher level, 99 point, I think it's 999 level of confidence that there's a 1 in a million chance of an infection or of a microbe surviving the process.
And I think you brought it to light very clearly in my mind that you can do log reductions on something, but that's just reductions from where you started. Mm-hmm. Tell us about how starting load matters and why the number of reductions that you might have to do might vary depending on that starting load.
Yeah, so there's different approaches to achieving sterility assurance level of 10 to the minus 6. So there's the bioburden or overkill method, as it's referred to sometimes. And so that's where you just assume a worst case scenario that you're going to have a 6 log level of contamination. And so you get your BI strips from your supplier and you test and you prove that the challenge organism, whichever is the hardest to kill for your sterilization technique, you can kill 6 logs of that microbe. And so in that scenario, you double the condition to get the sterility assurance level of 10 to the minus 6. In some instances, and one of the approaches that the agency is very open to is a bioburden method. So in that scenario, you actually categorize your bioburden. And so you look at what are the microbes that are part of your manufacturing process. You look at the levels, and so you test for them. And then what you show is that you're able to kill not only that particular organism, but that particular organism or those organisms at the level of your bioburden. So, for example, if you're in a manufacturing environment and you have a native bioburden of 1 log, so 10 microbes, the agency is going to say, we're not going to do as low as 1 or 2 logs. So there's kind of a minimum threshold of 3 logs of bioburden. So in that scenario, you'd say, okay, we have 3 logs that we have to reduce. And now to get to cell 10 to the minus 6, we have to add 6 more logs. So actually in that scenario, you're showing a 9 log reduction versus the overkill method where you're showing a 12 log reduction. And where that comes into play is that all of a sudden you can start reducing maybe the amount of additive that you need to use in your sterilization process. Maybe you reduce the amount of time. So for these more sensitive devices, you're able to use a less harsh condition that allows you to achieve terminal sterilization without negatively impacting the performance of your device.
That's really cool. So you don't just take this blind approach to it. You actually go in and say, what do we actually have here? And then how do we get to the level? And we're not making it any less sterile than we would have otherwise. We're just actually knowing where we started and knowing where we ended up.
Yeah, and there's a cost associated with that monitoring because as you are in your manufacturing process, you have to confirm with some level of frequency that your native bioburden and the amount of bioburden hasn't changed. And so if you use this technique, it's really a cost-benefit analysis of how much does that ongoing monitor cost us relative to the value of terminally sterilizing as opposed to aseptically manufacturing. Because even though aseptically manufacturing isn't as safe as terminal sterilization, it's significantly more expensive. And I've seen that aseptic manufacturing may cost up to 10% of your cost of goods sold, whereas terminal sterilization is in that 1% to 2% range.
So for products that are being aseptically manufactured, that may not actually be the sweet spot. They might really want a sterilization method that works with their product so that they can then actually reduce costs.
Absolutely.
Wow, that's fascinating. Well, speaking of methods, so at the end of July, FDA announced they recognized a new standard. And of course, as we've talked about on the show before, FDA maintains a consensus standards database, where they're not saying you have to do these things, but they're basically saying, if you follow this standard, which we recognize, then we're not going to ask you those questions. We're sort of trusting that that's a reasonable way to go about things. And not every standard's included in there. And this goes from everything from electrical safety standards to risk management to how you run quality systems and everything else. But they recognized at the end of July ISO 22441, which is a low temperature vaporized hydrogen peroxide method. Tony, tell us just a little bit more about this method, and then about this approach of recognized consensus standards for sterilization methods versus there's another way to do it. And I think you'll unpack those two for us.
Yeah, so the consensus standards are, really they are programs and efforts that happen over years. And so, for vaporized hydrogen peroxide, they've been pursuing that. And by meeting those standards and being able to set some of those standards, they're able to then go to the agency and say, here are the standards, we've met them. Can you move us from novel modality to established, and in this case, category B for vaporized hydrogen peroxide? And so that is one approach, and it has been the approach, right, up until 2022. So in May of 2022, the FDA started a new program as part of their efforts to support new sterilization techniques, a master file program. In 2022, it was a 510K master file program, where if you were sterilizing your 510K cleared device with ethylene oxide, you could actually use a sterilization company's master file program as a reference to move from EO to the new modality of your choice without going through a full FDA program or full FDA submission and a resubmission of your 510K.
Because oftentimes changing your sterilization method is going to kick you right back to, hey, submit a new 510K.
Yeah, absolutely. And so in addition to that program, so that was 22, in April of 23, so this year, the agency took it one step further. And so it's not just 510Ks. There's and I wrote it down, but it's the CDRH's master file pilot program for radiation. So it's the radiation pilot program. And that's, a little bit of a misnomer because it's moving from radiation or ethylene oxide. So it's not just radiation. But whereas the 22 program was for 510K cleared devices, this is for class 3 PMA designated devices. And so now you can move from a radiation or an EO modality to a alternative sterilization modality if that modality has a master file that's in place. And so what the agency has shared with us is that a way to move from a novel designation to an established designation would be to go through that master file program as an alternative to a consensus standard approach.
You're sort of giving a recipe for, hey, here's how this is going to be done. And then when some company references that, they look and go, oh, yeah, we do have that in our file, and off you go.
Absolutely.
Yep. It's really cool. I understand you guys might have a little bit of experience with this particular methodology. Tony, I would love it if you would just unpack your technology at NovaSterilis. Also how you're approaching, getting through these hurdles. And also, as we mentioned earlier, this FDA challenge grant that you are one of a very few recipients of, along with some big sterilization names. And so, again, congratulations. Tell us about whatever order you want, your technology, your process, and about these interactions with the FDA.
Yeah, so I'll start with the challenge grant program because it's really a fantastic program, and it's something that's moved our technology and our platform forward at an accelerated rate. So as I said earlier, the agency recognized there were some challenges with ethylene oxide sterilization. They put a challenge grant out there where they had a request to the marketplace for companies to propose their sterilization methodologies or new techniques, and to enter into the challenge grant program. And I shared with you as we talked a little bit before, Gene, is that originally it was a program we weren't going to partake in because there was no money associated with it. And so I'm looking at this grant saying, why are we going to do a grant and there's no money? But I will tell you, 3 years later, we were fortunate enough to be one of 4 companies selected as part of this program. Steris, Noxalizer, who's has a partnership with Sterigenics and Stryker and ourselves, is that with what I know now, if they had offered 500,000 instead of what we got, I would have turned the money down and taken what we got, because the interaction with the agency has been fantastic. It's been collaborative. It's been informative. It's really helped us move this modality. And as I said, last year there were 4 clearances using supercritical CO2 processing as part of their submission. And a large part of that was, the help from the agency to answer questions and be open to some submissions and information that we're able to provide. And so it was through that program that we were able to educate some of the key people at the agency on what our technology is about. And so, supercritical carbon dioxide, we joke, we're an overnight success. It's only taken 20 years of developing the technology and moving it forward. We were actually building upon a knowledge base that existed before. So supercritical CO2 processing has been around for 50 plus years. It's one of the way they decaffeinate coffee. It's the way they extract Taxol from yew trees. Which is, used as an oncology drug. And so there's a large body of knowledge around supercritical CO2 processing and the safety of supercritical CO2. We applied it to the medical arena and specifically sterilization, starting in about 2000. We were commercial in 2008, where we brought the first terminal sterilization process to the tissue banking industry, where we were able to not negatively impact the performance of tissue, whether it be bone or soft tissue like ACLs, ligaments, dermis, et cetera. But what is our modality? What's unique about our approach? So we are a low temperature terminal sterilization process. We are minimally reactive with the products that we're sterilizing, and our probably most unique calling card is we're deep penetrating because of the pressures at which we operate our process. We're able to penetrate something as dense as a femoral head, which is about as dense a material as you can have. Supercritical CO2 will penetrate to the center of that, and we're able to achieve sterility assurance levels of 10 to the minus 6 on a product like that. And the uniformity of the exposure is fantastic. And so we're able to sterilize complex devices that have multiple materials with that they're manufactured out. For example, an endoscope, 25 to 30 different materials. And we've shown the ability to be compatible with that variety of materials and still achieve terminal sterilization.
That's kind of incredible, especially on the complex surfaces, because honestly, how things are packaged even has always had an effect on sterilization methods. And I mean, how does that compare? Like you mentioned a femoral head. How does that compare to ETO? And perhaps also in the question of time, even if they could both do it, how do they compare with respect to time?
Yeah, so, ETOs are the workhorse, right? And I don't want to, throw too many darts. Obviously there is the environmental and health and safety risk, but ethylene oxide, has proven to be a real workhorse. Where we come in is if you have something again with that complex shape that you want to sterilize, we are not only the deepest penetrating, but we penetrate at the fastest rate of any of the different sterilization modalities when you look at the complexity of the device. And one of the things with supercritical CO2 is you don't have a post outgassing. So the ethylene oxide, you may have a 1 hour to 2 hour processing cycle and a 15 to 20 hour outgassing cycle. In our modality, you run that 1 hour, 2 hour, 4 hour cycle, and then when you're done and you depressurize the chamber, you're done. You're ready to ship. There isn't an outgassing component that's associated with it. One of the other, I think, when you start looking at as a manufacturer, how do you want to sterilize? And so there are a number of manufacturers who want to use contract sterilization facilities. And so something like an ethylene oxide or a gamma where there's a large CapEx associated with it and a lot of expertise to make sure that you're complying with OSHA standards and EPA standards, outsourcing that manufacturing makes a lot of sense. In our case, you have the ability to either outsource or bring that manufacturing or sterilization capability actually in-house and actually make it part of your inline processing. We are geared more towards the mid-range volume. So the really high volume applications, for example, we talked about Band-Aids in the beginning. It's not a good fit for supercritical CO2 sterilization because the chamber size. I mean, in ethylene oxide, there's chambers that you can drive a tractor trailer into and sterilize in its entirety. From a supercritical CO2 standpoint, there are no chambers of that size or of that volume capacity.
Wow, sounds like an absolutely fascinating technology. Do you have any? You want to share any cool stories or things that you guys have been proud of that you've achieved?
Yeah, so we have over the years sterilized. It's a broad array, everything from donor tissue from cadaver tissue used in surgeries, animal tissues, xenografts, to therapeutics, to spices. So it turns out vanilla beans and black peppercorns oftentimes are sterilized with EO and residual EO because of the way their shapes have dead pockets in there. And so we've had a couple of spice companies that have come and said, we're looking for an alternative to ethylene oxide sterilization for our spices. Bunny suits.
Wow. I mean, that's actually shocking to me that ETO is used for spices. I'm a little shocked actually, but wow.
I was too when we got the phone call. I did not know that. And so, there have been a number of devices that we've actually sterilized. And one of the other things, and a bit of a side note to this conversation, but supercritical CO2 is not only used for sterilization, but we actually use it with some of the tissue products that I've talked about. We're actually using it to decellularize. And so when you get, these tissues are coming in, you need to decellularize them. Oftentimes, some pretty harsh chemicals were being used, and supercritical CO2 is a great solvent for cleaning. And so we've been decellularizing tissue. And there were some really neat projects at universities. University of Arizona did a project where they decellularized spinach leaves and then sterilized it with our process, did both processes with our process, and then used the fiber backbone of the spinach leaves as a template for creating vascular mats, seeding those stems of the spinach leaf with stem cells and creating vascular mats to make a model of for vascular drug delivery to anaerobic tumors. So some really cool stuff that we've seen
all
over
across
time.
The board.
Creative minds out there using the technology in ways we actually never envisioned when we started.
Well, that's so cool when you get a platform and it works for so many things and has, similarities too, but then differences from established ways. You can really play with some cool stuff. Who should be coming to you, Tony? Who if they're listening to this video and they're like, wow, this is really educational. I'm thankful for all this. What sorts of people should be picking up a phone and giving you a call or emailing you or connecting however you tell us to?
Yeah, so whether it's email or call, we're still old school. We answer the phone. So we love talking to people about what their needs are. And so at the end of the day, if someone has a product that they're developing and they want to understand what their sterilization options are, we have a proof of concept program that we run that's kind of down and dirty, simple, that people can send us their product or their materials that they're going to use in a product just to see if they're compatible. And whether it's our modality or anyone else's, I've been in the medical device world for a long time now. And what I will tell you is you bring new products to market or you start designing, which Gene, a lot of people don't think about sterilization to the end, and you assume that something's going to work. And oftentimes we see that people have to go and redesign materials being used or redesign configurations because sterilization became a roadblock. And so I would really challenge people that are in the earliest stages to do some compatibility testing of your material with whatever modality. And we have people, they're going to do ETO and gamma and us and vaporized hydrogen peroxide. And I look at those as the people who are doing it the right way, understand what the compatibility is, so when you build it, you know which direction you're going to go later down the line. Someone who's got a device that they're concerned about the ETO supply chain, and they want to come to us and just talk about what an insurance policy looks like for proving out that this approach works should something happen with their current approach. And then oftentimes people who are doing biologically oriented or biologics, because there's now a lot of terminal sterilization modalities that are compatible with biologics. And although we're not compatible with all biologics, we've got a pretty broad effort that we've undertaken to show our compatibility with a number of biologics, whether they're therapeutics or scaffolds used for delivering drugs.
That's kind of amazing too, because you think about it, the point of sterilization is to remove biologics. So when you find a sterilization method that's balanced enough that it can retain what you're trying to retain and kill what you're trying to kill, I think that's a pretty fun niche to be in.
It's been great, and obviously we're learning a lot, and we've been doing this for 20 plus years at this point, and we're still learning as to what else we can do with the platform to not only sterilize and clean and decellularize, but the new frontier is going to be functionalize. And so there is some approach using different additives where you can functionalize materials for specific applications.
Very, very cool. Tony Eisenhut, CEO and co-founder of NovaSterilis. I'm thankful to our mutual friend Kelly Sycon for putting us in touch. And Tony, I'm so thankful for you spending the time with us today to talk about this. Very, very educational. I think people are really going to enjoy this.
I appreciate it, Gene. Great. Thanks for taking the time, to you and Steve, to spend some time with me this morning. It's fantastic.
We appreciate it. And for everyone listening, as always, we want MedTech Crossroads to be education that saves you time on your MedTech development journey. And I think Tony just did that today. So thanks again, Tony.
Absolutely. Have a great day.
What a great time with Tony. I think he's going to be back. He's what a great resource for sterilization. Really appreciate him. Coming up last, upcoming events in the local and national ecosystem, brought to you from all the places we can call as we look for what's happening this fall. not only in the Midwest but beyond. But coming up next week, August 30th, Cleveland, Ohio, the Midwest Showcase put on by Project MedTech. I will be there Wednesday in Cleveland. Hope to catch up with you there. September 7th, the Women in MedTech Forum in Massachusetts. That's powered by Mass Medic. Always great events. Celebrate Invention is a gathering of the local ecosystem, the local Michigan ecosystem, on the Michigan Union's second floor. This is always a great yearly event. September 14th, 2023, coming up fast. Resi Boston, if you're an investor or you're a startup looking for investors, this is the place to go. The deadline has passed, but you can still go to Resi. We have an old slide up here, but Resi is still happening September 18th through 20th in Boston. The 2023 Michigan Life Sciences Showcase put on by MichBio coming up September 18th in Lansing, Michigan. Drug Discovery Symposium. They're looking for posters for that. If you are a researcher for drug discovery, they would like to hear from you, michbio.org, because they're looking for a poster session for the Drug Discovery Symposium. Medical Device Summit, the Digital Health Forum, the Cannabis Science Symposium, and the Patient Advocacy Forum. Dedicated tracks for each September 18th. The A2 Biosocial is coming back. Paleo Rooftop. I tell you, the last installment of this, Matthew Himes and Company, wow. I was there. It was amazing. 200 people, maybe? I mean, just kind of going by gut. It was large. So what a wonderful place to be. Wednesday, September 20th, 2023, 5 to 7 p.m., sponsored by Spark, Nucleate, and part of the A2 Tech 360 week. The MedTech Conference coming back to Anaheim, October 9th through 11th, the MedTech Conference. And then going to Texas, October 25th through 26th, Project MedTech is hosting the Startup Symposium. Also want to remind you of the Eye and the Chip coming October 8th through 10th at the Southfield Weston Hotel. Synthetic Vision. Very, very cool. Dr. Hesberg and company there. And then coming in November to Cincinnati, Ohio, the Artificial Intelligence Summit. AFTO and RAPS taking over the old Xavier conferences. This is the AI conference. Earlier this year was MedCon. This fall is the AI Summit. And then in a moment, I'll tell you about the Combination Products Summit coming up, but the AI Summit is November 14th through 16th. Just found out about this, November 20th through 21st in Orlando, Florida, the American Conference on Clinical Trials and Regulation of Medical Devices. Anyone who's been part of a medical device development effort knows how hard it is to get through a clinical trial. Glad some attention is being focused there. And then back to AFTO RAPS, this time in Fort Worth, Texas, November 28th through 30th, co-sponsored by the FDA, the Combination Products Summit put on by AFTO and RAPS. If you have any questions, comments, contact at medtechcrossroads.com. Or if you're watching on the Zoom link, you are always welcome to raise your hand and let us know if you have a question or a comment, an upcoming event that we missed and you think everyone should know about, or a recommendation for something we should be talking about on the show. We're always happy to hear from you. And while I give just a few moments for that, we've really been taking off on YouTube. The live shows continue always. It's such a fun way to do things. And you don't get to redo them, which I just love, because otherwise I would do these things like 10 times. So it is what it is, and people are watching also on YouTube. We're really finding the growing niche there as well, so that's wonderful. You can watch the show live on Zoom every Friday at 2 p.m., or you can watch it live on YouTube, or you can watch it anytime you want on YouTube. And we break up the live shows also into segments that are now topical, so it's very fun to see that finally happening. We'll just take a quick look here. If there's any questions or comments, not seeing any, I want to thank you for being here with us on MedTech Crossroads. It's always great to see you. We'll see you next week.